This is episode 1 of the Immunology podcast, merging stem cells with immunology with Dr. Filipe Pereira. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome to the first episode of the Immunology podcast, where we have conversations with immunologists. We are very excited to be launching this podcast, and we have some great guests lined up over the next few months, including Drs. Carls June at the University of Pennsylvania, Judy Lieberman at Harvard Medical School, and Bernardo Franklin at the University of Bonn. If you'd like to learn more about the Immunology podcast and upcoming guests, visit our website at www.immunologypodcast.com or follow us on Twitter at @immunopodcast. Today we have Dr. Filipe Pereira from Lund University on the podcast to talk about his research understanding hematopoietic and immune cell identities and translating these findings into novel gene and cell therapies. We've also got our usual roundup of recent highlights in immunology coming up. But first,
are you using dendritic cells for your research? With the Immunocal Dendritic Cell Culture Kit from STEMCELL Technologies, you can generate immature as well as mature dendritic cells from human monocytes. Learn more at www.stemcell.com/DC Culture Kit.
all right, it's roundup time. How are you today, Brenda?
Oh, I'm doing great. Very excited about our first roundup of the season.
Excellent. Yeah, so am I. And so I guess we're going to start here, dive in. The first paper is called Expansible Residence Decentralizes Immune Homeostasis. This paper is by Sathe Wajising. It's a very interesting paper here. It came out on the 17th of March in Nature. And it really kind of does a very impressive deep dive into the role of resident T cells. And it's an impressive tour de force of work because some of their experiments go up to 450 days. And they use a large number of mouse models. They do parabiosis experiments. They do intravital microscopy. It's insane in a good way, but very, very impressive. And so what this paper really does is it tries to establish kind of the half-life and kinetics and homeostasis of resident T cells. So if we think about T cells generally, we have, our naive T cells, and then we have our memory T cells, and then we have these resident T cells that kind of stay in the tissue. And so they knew that resident T cells typically don't require TCR stimulation, but these regular memory cells don't need TCR-dependent signaling to survive. But these resident memory T cells, the ones that sit in the tissue, look and have a lot of properties like they have undergone recent TCR stimulation. So they kind of want to understand what these cells do after they're formed. So if you form these resident tissue cells, do they sit in the tissue for a long time? Do they move in and out of the lymph and blood compartments? Do they move between tissues? So on and so forth. So they used a bunch of fancy mouse systems. It's kind of hard to describe just on a podcast, but they use a system known as the P14 system. And this is a pre-recombined TCR that will respond to a specific virus, the lymphocytic choriomeningitis virus, LCMV. And so these are P14 immune chimeric mice, and then they do this on top of CD45.1 or Thi 1.1 to be able to track host versus donor immune cells. And so they use this as a system to have a preset antigenic response and then see what happens with these resident cells once they have been formed. And so then they do a host of experiments, just looking at how long they sit in various tissues. They do parabiosis experiments, where they stitch two mice's circulation together to get your double Frankenmouse. And then they try to see if the mouse that had the resident tissue or not, which one that had been kind of primed, if immune cells move from one mouse to the other. And so through this, they show that the resident tissue T cells really just stay where they are. There's some exceptions, notably the small bowel seems to be more in homeostasis with the circulation or the other mouse in these studies. But largely, these T cells just sit there. They don't leave. They don't change. They're ready to be primed and activated again. If you do another antigenic exposure to the same antigen, they don't get it. It's not like because they were already there, they're not going to respond anymore. They actually go at it hard and respond appropriately and expand further.
So a question I have is that we already mostly knew that resident T cells are a very stable population in the tissues that they inhabit. What is the angle that this paper has that really makes the difference?
So they track it for hundreds of days and show that they never leave. It's not like they're being generated by continual stimulation and then move to the tissue and then die eventually. They just sit there forever. And then what they show is then they take these, there's this new model that I hadn't heard about before where you take mice from a pet shop and you co-incubate them with mice from our SPF conditions or standard pathogen-free conditions or specific pathogen-free conditions to like have all this massive antigenic load from the real world come. And in this case, what they're showing is that when you have that antigenic load, it's not like the existing resident cells don't get thrown out and then new ones come in for the new pathogens. They just expand the population. That wasn't known either. There had been some thought that you could only have so many T cells in the tissue at a time, and that you're kind of capped, and so that there would be an exchange as new threats and challenges come. And that's not actually the case. They just straight up add more. And so it shows that you can do this co-culture, co-incubation system with these pet store mice, and you just double down and add more immune cells. And then they also identify the half-life in the various tissues. The female track was one of the more sturdy. So the uterus and vagina didn't shift when you added in these other pathogens from the pet shop mice. They actually just doubled down on their own antigenic load that they already had. But the small intestine would drop down and really start responding to the new system, which kind of shows you, being my favorite organ, of course, that the gut is much more sensitive to the microbiota status of whatever it's being assaulted with at a time. And so you can imagine these pet shop mice have all these other microbiota come in, and the gut has to have a much more localized tissue response than everything else potentially. And so those were where they'd see a shift. And then obviously they showed the normal shift that we'd expect in the lymph tissue. And so there'd be homeostasis there, and then a changeover in the responses there. So it was really just an impressive tour de force of understanding the half-life and the persistence of resident cells versus memory cells versus just lymphoid floating cells in using the T cells as a model system in this case to really understand this process. And it's actually pretty impressive how long it lasts.
Nice. Thanks. I have 2 words for you, Jason.
I'm listening.
The tumor microbiome.
Ooh, I like this.
So for some time, it has been described that there are bacteria that colonize and live within human tumors that are found in tumors from patients, and that not only do they live there, but also they correlate different strains of bacteria seem to correlate with different types of tumors. And there's a lot of work done, for example, places such as the Weizmann Institute in Israel. And the paper I want to talk about today actually comes from the lab of Jardinia Samuels from the Weizmann Institute. And it's called Identification of Bacteria-Derived HLA-Bound Peptides in Melanoma. It was published in Nature in the same issue as the paper you presented on the 17th of March. And the first authors are Shelley Klaura and Adi Nagler as co-first authors. And in this paper, they looked at different metastases from 9 patients, and they actually found several unique HLA1 and HLA2 peptides that were derived from bacterial proteomes. And they identified overall 41 species of bacteria in this samples, and they in fact show that there are bacteria living inside the melanoma tumors, and both melanoma cells and the immune cells present peptides derived from this bacteria in their HLA proteins, which I thought was really mind-blowing. Have you heard of bacteria living inside of tumors before?
So I hadn't known that part of it. I mean, I think I remember one little thing about it, but not how they get there. But what's interesting about this is there's this whole movement now to understand the role of the microbiome in immuno-oncology, to the point that certain microbiota can affect the metabolism of drugs and make them more or less effective for chemotherapies or immune checkpoints. But also the microbiome seems to prime, and different compositions can be either good or bad to prime the immune response against tumors. And so they're finding that germ-free mice or mice with a response to a checkpoint inhibitor versus those without, these different types of mice, these different types of statuses of the microbiome actually affect the ability of the host immune system to respond to tumors. And so it seems like the bugs prime it, but I didn't know that there were bugs growing in tumors, and how they got there, I have zero idea. And then now they're presenting antigen as well. Then that leads to a whole bunch of questions. Like, can you target them? Which is what it sounds like they're getting at here.
Yeah, so they identified this several peptides. And also, interestingly, they seem to find specific HLA alleles that were most likely to be presenting bacterial peptides that also are more hydrophobic than endogenous peptide in general, which seems to correlate with the reason why they bind better to these HLA alleles. And they indeed find that certain peptides, they could indeed find T cells that could respond to these peptides being presented that were derived from the melanoma samples from these patients. So in summary, what they do is they identify a myriad of bacterial species living in these tumor samples. They show, using several microscopy techniques and also fishing the 16S RNA sequences from samples, and they show that the bacteria are literally inside the cells. They show that both melanoma cell lines, so tumor cell lines, and antigen-presenting cell lines, when co-cultured with this bacteria, can take bacteria in and present its peptides on its surface, on their own MHC molecules, and that some of these peptides can be immunogenic in T cells derived from the patients where they come from. So I think that in general is super interesting how the results from this paper, and whether this can have some usefulness or some impact in treatment is still to be said, because it's very new, and I don't know how many of these peptides would actually be presented to enough levels to be actually good targets. But I think in general, as a proof of principle, is really, really interesting.
Oh, that's fascinating. I wonder if that could then lead to some cell-based therapies with chimeric antigen receptors that are targeted against these bacterial peptides, since they're obviously going to be somewhat unique compared to the host.
Yeah, so we'll see what the future brings in tumor microbiome research.
Indeed. All right. Well, I guess it's my turn for the next one. Continuing on the theme here, I'm going to stay in the gut because it's, of course, the best organ and is, an important immune organ in all of itself. And this is a very interesting immunometabolism paper out in Science on March 19th by Sullivan et al. And it's titled Gamma Delta T Cells Regulate the Intestinal Response to Nutrient Sensing. And this was very fascinating. So what they basically did is they had two diets, a high protein and a high carbohydrate diet. And they showed in step 1 that gut cells, enterocytes, responded to the high carbohydrate diet by shifting in a couple of ways. They upregulated genes that would handle higher levels of carbohydrates for metabolism. That's not super surprising. But then they also demonstrated that this was associated with a shift towards stem cells and transient amplifying cells in a change differentiation program, demonstrating that the gut was responding and shifting to cell types, transient amplifying stem cells and stem cells that preferentially consume glucose. And then they identified from there, but they kind of took it a next step. And this was actually the group that did intravital microscopy. I realized that I had screwed up when talking about my other paper. They didn't do intravital, but this group did. And they found that intraepithelial lymphocytes and lamina propria lymphocytes swarmed to the base of the crypts and swarmed to the tissue in response to sugar, and actually moved there. And then they followed the signaling cascade and figured out that there was gamma delta T cells that regulate this carbohydrate transcriptional program. And so through a series of elegant experiments using antibodies, depletion strategies, and knockouts of the various cell populations, they identify specifically it's these cells that are doing it. And they are doing it by regulating innate lymphocyte 3 or ILC3 secretion of IL-22. And so in the presence of sugar, these gamma delta T cells tell ILC3s in a mechanism that they haven't figured out yet to downregulate IL-22. And they show that the IL-22 suppresses this program at baseline, which is really interesting because IL-22 is also known as a driver of STAT3 activation and general wound healing. And so there's a lot of interesting crosstalk here. And then in a series of experiments, and part of this, they show that this program persists for quite a while and takes time to shift off. And then they also identify that this program, through lamina propria signaling of the gamma delta T cells, is going through JAG2, which is a Notch ligand. And so it's upregulating Notch-based signaling, Notch-Wnt pathways, which are the molecular switch that drives this, de-differentiated state. So you have higher Notch-Wnt when you're in a high glucose state. And so it's an incredibly elegant set of experiments. I was really impressed with it and how they really were able to dissect all the way down to the immune pathways what's going on. And of course, it does, in a good way, leave some extra work to do so they can get their follow-up funding and papers out on that linkage between the gamma delta T cells and the ILCs. But it's like a very nice set of studies. They show that it may be cyclooxygenase dependent in tough cells as well as part of the regulatory pathway. But they don't have a better sense quite yet of that linkage between the gamma delta and the ILCs. But then they show the ILCs directly through IL-22 activity or what suppresses this pathway at baseline. So it's a very interesting study. It's right up my field of diet microbiome and immunology and intestinal differentiation. It has all of my favorite food groups at once, pun intended, hashtag dad joke. And I thought it was fascinating. And it's actually been kind of making the social media rounds as well. I've seen it on a couple of spots on Twitter and being pushed already. It's pretty cool.
So I have two questions for you. Regarding the role of the gamma delta T cells, how do they show that they are crucial for this cascade of events? And do they dive a little bit deeper on how do they sense the change in diet?
So the first part, yes. The second part, no. So for the first part, they do gamma delta knockout versus alpha beta T cell knockouts. They do also antibody depletion as a first pass and show that it's dependent on that. They show that if you just do enteroid culture and do these, it doesn't work. But if you co-culture with immune cells, I think it was a co-culture. I have to dig back in. But they did a couple things where they either recapitulated the downstream signaling through like the IL-22 inhibiting it. They also did the ligands, I believe, and I think they at one point did a co-culture as well, and demonstrated that it was the gamma delta T. I think they really locked that in pretty well between the knockouts and the germ-free studies, and they did a bunch and really demonstrated that it was those. But I don't think they didn't explain how the immune cells are sensing the sugar. I mean, we know that the diet is going to be in homeostasis of the blood, so you're going to have a different blood glucose level, but they don't go into how the gamma delta T cells are doing something to the ILCs or what program that is. And they couldn't find a good cytokine signal outside of IL-22 to begin with. And so I don't think they have dissected that part of it yet.
Very nice. Wrapping up, I have one more paper to share with you. And this paper is all about my favorite kamikaze cells of the immune system. Of course, those are the neutrophils. And how neutrophils can activate antigen-presenting cells and monocytes by releasing their famous nets, their neutrophil extracellular traps. So this paper is titled "The Cytosolic DNA Sensor CGAS Recognizes Neutrophil Extracellular Traps," and kind of gives the whole thing away, I have to say. First author, Falco Apple from the lab of Arturo Siklinski at the Charité Institute in Berlin, so one from Germany. And in this paper, they try to address the fact that we know that, as you know, neutrophils, when they can get activated, when they are activated by they recognize microbes or in the bloodstream, or they're coming in contact with molecular danger signals, they get activated. And one of the things they do as a really kind of large offensive is they release this so-called nets, which are a mix of chromatin. So they really kill themselves and they release their chromatin, with proteins. And one of them is the neutrophil elastase, which is characteristic of these nets, and peroxidases and other lot of and this mix of DNA and proteins is really good at immobilizing pathogens, at kind of sticking them together, making them easier to degrade, to pick up by phagocytosis. And the thing that they also know is that these nets can activate myeloid cells, dendritic cells, monocytes, and but it's not clear what is the mechanism. Until now wasn't really clear which was the mechanism that the nets did this. And so basically what the authors did is, in a series of very elegant experiments, they progressively look at the activated neutrophils in vitro, and they show that when we're together with monocytes or myeloid cells, they show that the release of these nets induces the expression of type 1 interference. And so when they co-culture them with PBMCs, and they slowly kind of tear apart all of the components of the nets, and they come to the conclusion that it is really the DNA in this nets, and not only the DNA itself, but the DNA as after being degraded by DNases. DNase 1 is this extracellular DNase that commonly will degrade these nets that at some point need to be taken care of and need to be degraded and taken out of the way. And it is this DNA sequence, or these DNA fragments, that activate a very known DNA sensor pathway, which is cGAS-STING, which downstream of this pathway is the expression of type 1 interference. And I think what I liked about this paper was that they really looked into very closely at all of the different possibilities. They looked they do many experiments in which they inhibit cGAS, they inhibit STING, they also inhibit the effects of other components of the nets to prove that it is really the DNA that is doing the job, and it's really the activation of cGAS and STING that really is driving this activation and this interferon production. And what I also think they did, which is really, really nice, is they have this beautiful videos, which we should hopefully post into the show notes, of activated neutrophils released in their nets, and macrophages and so monocytes coming and phagocytosing the DNA and the nets outside. And also what they look at, which is very important, is the fact that cGAS can only be activated by DNA in the cytosol. So one of the questions they had is how do the net get in contact with cytosolic cGAS? And for that, they show that what happens is that the nets are partially degraded by DNase and outside in the extracellular environment, and then they're taken up by phagocytosis. And then they also showed that one of the proteins that is part of the net, which is the neutrophil elastase, can somehow mediate the release of the DNA into the intracellular cytosolic space, where it can actually activate cGAS.
You would say that like the big impact of this is a lot of the molecular mapping hadn't been done before, so we kind of knew the broad brush strokes of this process, but not the molecular pathways and down to being able to watch it happen and identify what was happening at each point along the way.
Yeah, I think that is basically they show that what exactly is the molecular mechanism that connects the net being released by the neutrophil and the production of type 1 interferon in the myeloid cells that are around the neutrophil. And of course, this has always activation of these cells always is very important for stimulation of the immune response. So I think it's really good to understand how this can happen, and also the function of this neutrophil elastase into translocating the nets from the phagosome into the cytosol was not really, I don't think had been really characterized before.
Awesome. That's super fascinating. All right. Well, I think we're out of time for this set of roundups today, but I'm sure we'll be back at it soon with the next set. Moving on, we're going to be speaking with Dr. Filipe Pereira at Lund University. But before we get to that, are you interested in differentiating human pluripotent stem cells into monocytes? The STEMDiff monocyte kit from STEMCELL Technologies generates millions of monocytes ready for downstream assays or for further development to macrophages or dendritic cells. Learn more at www.stemcell.com/monocyte kit.
Today, we're talking to Filipe Pereira, who is an associate professor at the Faculty of Medicine of the University of Lund. His lab at the Lund Stem Cell Center does amazing work studying the differentiation of hematopoietic cells and ways of reprogramming cells towards a hematopoietic lineage, which has exciting applications in regenerative medicine and immunotherapy. Based on this research, Dr. Pereira has also co-founded a company, Asgard Therapeutics, which aims at reprogramming tumor cells into antigen-presenting cells that can effectively activate the immune system. Dr. Pereira, thank you so much for joining us today.
Thank you so much for the invitation.
It's a pleasure to have you here. So, as I mentioned, your lab has great expertise in immune cell reprogramming. And not too long ago, you published a very, very nice paper in Science Immunology that's titled Direct Reprogramming of Fibroblasts into Antigen-Presenting Dendritic Cells. I think it's a fascinating concept. But your journey with reprogramming fibroblasts into hematopoietic cells has been working on for a long time. So I was hoping you can maybe tell our listeners a little bit how you got started with this field and what is your lab doing at the moment in this area.
Sure. I think my journey in reprogramming has started quite a while ago, 17 years ago. I did my PhD in London. The question in this field was that whether or not we could take a somatic cell back to the pluripotent state. So we were using cell fusion before Shinya Manaka came out with these 4 transcription factors that allow us to induce pluripotent stem cells. So we were fusing lymphocytes to embryonic stem cells to study the mechanisms that would allow us to impose the pluripotent state. So then I moved to New York for my postdoc, where I started bringing in these reprogramming concepts to hematopoiesis. And we set out to identify the transcription factors that would induce an adult somatic stem cell for the first time, an hematopoietic stem cell. So what we got was this recapitulation of the process of hematopoietic stem cell generation in vitro with the transcription factors GATA2, FOS, and GFI1B. So then in 2015, when I started my own independent lab, I was motivated by the idea to bring these reprogramming concepts that have been working for a long time to the field of immunology. And we got interested with the dendritic cells because of their fantastic antigen presentation, capacity for antigen presentation and so on. And then we expanded and now are actually interested in many other cells, but always touching the reprogramming as a way to inform us in this journey. So the lab now has 3 main areas, all kind of having this the core of bringing reprogramming and immunology together. And the first one is to actually use direct reprogramming to understand the transcription factors that initiate and impose immune cell fates. second line, we try to use this in different ways to treat cancer, right? So you mentioned that we apply these networks that we identify, and then we're trying to turn cancer cells into efficient antigen presenting cells, so to counteract the tumor evasion mechanisms. And then we're so interested in the more basic mechanisms of transcription factor engagement and how this process goes through the development of the immune system, since the generation of the hematopoietic stem cells towards the mature cell identities.
So I've always been really curious since reading your work about why fibroblasts become dendritic cells. I think almost from an evolutionary biology perspective, how and why has our bodies evolved the ability to go from a fibroblast to a dendritic cell? It seems, at least at first glance, we need to do some thinking about that. I wonder if you had any thoughts about why this program now exists, what's the evolutionary advantage, what functions happen if you, say, ablate it, and do you see some gross dysfunction health-wise that would explain why it's important?
I think it's fascinating, right? That every single cell in our body still retains the capacity to become in anything else, right? But it's somehow restricted. The whole set of genes are there, right? And then they are restricted by epigenetic mechanisms that ensure that when the cell divides, lymphocytes keeps being a lymphocyte and not become a fibroblast, for example, right? So, and it's the field started in the beginning of last century, actually trying to address this question, right? The field of reprogramming was trying to address whether cells would lose genes when they would commit into certain lineages. And the seminal work by John Gordon has shown us that they do not lose genes, at least the majority of the cells. They retain their ability, the plasticity and the ability to generate the whole organisms. So in terms of fibroblasts, right, we use fibroblasts as almost like a screening platform because they are easy to isolate, relatively homogeneous cell type that we can we can use tools very easily from reporter mice that have a gene that are selectively expressed in a certain immune cell type, and we use fibroblasts as this like tabula rasa cell type where we start from to impose immune cell identity.
So the fibroblast not necessarily 100% physiological, but is a good model system, in other words?
Yes, so it's a good way to start for us to identify these transcription factors that are key to elicit the process. But for example, in the case of the dendritic cell reprogramming process, where we identified P1RF8 and path3 from mouse fibroblasts, so it's shown that this network it's conserved in human embryonic fibroblasts or in dermal fibroblasts, in mesenchymal stem cells, and mouse and human cancer types. So I think in total, there are more than 50 cell types that we have shown that the process is conserved. This is still not published, but it's the work that is ongoing in my lab.
And I guess my last follow-up before I pass the baton back to Brenda is how biologically, so I guess what processes induce this in vivo and normal host biology? I ask this because I come from the intestinal stem cell field to an extent as well, and we found that adult cells reprogram through a fetal-like process during regeneration, and so this whole fetal program is actually used after injury in adult cells that de-differentiate and heal. And so I'm wondering, you're finding these conserved programs, where are you seeing it pop up in biological processes? Obviously not regeneration, but say inflammation, infection, other scenarios where you're seeing this reprogramming now occur.
Yeah, so it's an interesting point to think about how flexible the cell types are in the organism, right? So is it as flexible or as stable as we think they are, or is it just a much more dynamic system? So actually we don't know, and it's actually difficult to understand and to track this in vivo, because once the cell gets reprogrammed, then it becomes indistinguishable from the other cells. So when there are cases where in the case of disease, for example, there are some certain cancers called metaplasias that they're characterized by patches of intestine in the stomach or the reverse, and it's believed that they emerge through the reprogramming of the epithelium, as exposed to stressors. But it's also still not very clear whether what the molecular mechanism that initiate these conversions in vivo. So I guess we need to have better tools to understand these cell fate transitions in vivo. But we can we can speculate that would be reactivation of transcription factors or the erasure of the epigenetic mechanisms that are maintaining the identity of the cell, and these are probably influenced by pathological conditions like inflammation or chronic stress, for example. But it's still very open question at this moment.
Are you guys able to lineage trace some of the factors you're looking at, or is that still a difficult problem? I know in intestine land, they've been lucky to get some lineage traces, but that may be a fluke of timing and what's been able to come out. I don't know if you guys are having that same hurdle, if you've been able to, surpass that or not.
Yeah. So it is so far we in my lab we focus more on the in vitro reprogramming, and we use the in vivo reprogramming as a way to counteract the tumor evasion mechanism. So whether such a lineage conversion can within the dendritic cell lineage can occur in vivo, we don't know.
What I was also thinking about, I think the implications for reprogramming cells into, for example, antigen-presenting cells, I think are very clear from the therapeutic perspective. Maybe we can talk about that in a moment. But first, I also think what is fascinating is the idea of using reprogramming and playing around with the factors that you use to understand how it happens in the real situation. I think by maybe, do you have any stories or any particular examples that you can share about how using these techniques helps us understand what makes the identity of, in this case, immune cells in particular?
Yes. So for the induction of immunogenic endothelium with these other factors, GATA2, FOS, and G501B. So we also were interested in using this system, particularly in the human, as a model for the developmental specification of hematopoietic stem cells. So it's very hard to access that in the embryo, right? In the human embryo, it's very hard to study this process. So we wonder whether, because we can recapitulate that in vitro with the forced expression of these three transcription factors. So if we can understand a little bit more about this process and how these factors do it. So what we have done was in the beginning of the reprogramming, so we mapped the initial engagement of these factors and their interdependencies to the chromatin. So our idea was, let's see where they bind and if they need each other to bind to the target sites, and what are the genes that they are binding to in the first place, and whether this would be important genes for hematopoiesis. So what we found was that, interestingly, there was one factor, GATA2. So we did chip sequencing in the beginning of reprogramming that. So this factor binds to the majority of the targets, so it's a dominant transcription factor, and it does so independently of the others. But the other two, GFR1B and FOS, require the presence of GATA2 to to be recruited to the target sites. So we kind of uncovered the cooperation between the three transcription factors to elicit this immunogenic program, but also the critical role of GATA2 in leading the way towards the program, and kind of capturing the other factors into this complex that allows to kickstart the process. So now we are interested, of course, now with the dendritic cells and the multiple types of dendritic cells that we are generating, whether this is also the case here. Do they need cooperative binding, and are there transcription factors that are more important than others to kickstart this process, or not?
Seems like a great system to look into this part of the development that is so hard to access otherwise. And so maybe could you talk to us a little bit more about this idea that you are developing of using reprogramming tumor cells to kind of as a little horse you call it a Trojan horse approach to present antigens to the immune system. And a little bit about this is very interesting that you are going forward into bringing it to the clinic, and that sounds fascinating.
Yeah, so it evolved. The idea evolved organically, let's say, and we didn't thought about this in the beginning when we started the project. We were driven by the idea to bring these two concepts together. So this reprogramming and immune responses, right? So, but then during the course of the project, we thought, well, if you can reprogram fibroblasts from this species, maybe we can reprogram cancer cells and force them to present their neoantigens, right? So the ones that they acquire during the tumor formation, and they are hiding from the immune system with tumor evasion mechanism. So, for example, with the downregulation of MHC molecules. So that we started like that, and then we then would develop this idea because some of the people in my lab were very interested in innovation and starting new ventures. So we also start thinking about what would be the easiest way or the most attractive way to get to develop this into a product. And so then this kind of matured from a cell therapy into a gene therapy, because with a gene therapy where we could deliver the three factors inside the tumors would be off-the-shelf therapy. So the same viral vector would fit everyone, but at the same time we keep the personalized nature of the response, because the neoantigens would be patient and cancer specific. So yes, I guess that was why we got into this idea, but also a little bit of luck, because when we started this project, we screened for 18 transcription factors expressed in dendritic cells, and the type of dendritic cell that we got with these 3 hits were dendritic cells type 1 that are being implicated in these recent years to be critical for eliciting cytotoxic responses in the tumors, and they're necessary inside the tumors and also migrating to lymph nodes to kickstart a response against cancer. So yes, so we thought, well, those are the ideal cells. They are very rare in the peripheral blood. They are very rare in tumors. So if we can reinstate them by reprogramming the cancer cells or the other cells in the vicinity of the tumor with this approach, this would be revolutionary. So that's what we are aiming towards.
And you also mentioned that you're aiming at these cancer cells to present neoantigens to the immune system. But we know that also tumor cells express other tumor-associated antigens that could potentially be targets of the immune system. But why are you focusing on neoantigens? And what kind of data do you have to support that idea?
So far we have model antigen data. So we have cells that are expressing endogenously a model antigen, and we know that after they get reprogrammed in vitro, they become able to present that antigen and elicit CD8 T cell activation and proliferation. So we don't really, we're not really focusing on the neoantigens. Just the whole overall idea is that the cell starts to be become immunogenic and not evasive, right? So, but we think that the chances for these cells then to present these neoantigens that were hidden from the immune system, so after reprogramming, will be much higher, and we don't need to know which neoantigens are there, right? Which could also be a challenging topic to identify and distinguish these neoantigens from the tumor antigens.
Gotcha. So given that I think that's a really strong advantage of what you have, is you don't have to worry about any given neoantigen to replicate. And it sounds like if you just turn on, this approach is just turning on some DCs. What are your plans on how to then selectively or not convert too many random cells in your body to be DC1s? Or are you going to try to have the gene therapy vector go towards the cancer and do something kind of in that area, but you don't care what? How are you going to narrow this reprogramming of anything that it touches into a DC1 to be, not so I don't have DC1s lighting up in my heart and my lungs and my gut and, causing a giant cytokine storm, but they are activating, in cancer land?
Yeah, so totally agree with you. And the delivery is the problem here, right? And it's not only for us, but for many other groups attempting to develop in vivo gene therapies. So what we are doing now is kind of diversifying the ways we are attempting to deliver the three factors in vivo, which then will open many different opportunities. So we are using lentivirus, but also now starting to test adenovirus and RNA, so that we could, in some of the cases, try to adapt this system for an IV delivery, where we'll be not so limited by the intratumoral injections. But so far, we're pursuing an intratumoral injection of a lentiviral vector, which is relatively big and does not spread that much through the body.
So besides working on reprogramming dendritic cells, is there other research in your lab that you would like to maybe share with us, or some other new ideas that you guys are working on?
So, I have this ambition of being able to program every single immune cell and to map the transcription factor networks that control every single decision along the way, right? But so we are kind of trying to find more efficient ways to identifying these reprogramming transcription factors, and using the myeloid lineage first, but also doing the first steps into the lymphoid lineage, because this is a territory that is completely unexplored, and no one has come up with with this set of transcription factors that would program a T cell, for example. So so we are interested about that, and having more efficient ways to screen and to induce in these cell types.
Do you have any idea why the lymphoid lineages have been so hard to sort out? In a way, like, the B cells are highly plastic and they become eventually, these permanent plasma cells and such. But at least in, at a high level framework, they have a lot of, I don't want to say pluripotency, but they have a lot of lineage steps that are known that they go through. But I agree, like the reprogramming has seemed to be very poorly understood? You think it's a feature of things like a B cell where they should, air quotes here, but kind of have been evolved to work in one direction and just become more and more specific to an antigen and then preserve? Or do you think it's that we've just had poor tools? Maybe it's both. If you could comment on that.
So what I think is that, so, the cells mature in different places. They need to go, right, different anatomic places to get different signals. And it's complicated to recapitulate that in vitro. But on the other hand, I don't think anyone has really tried. So, it doesn't mean that it's hard, so that's what we want to try, because I don't know how much this was being attempted before, right? So, but yes, it's more complicated than the myeloid cell, I would say.
I wonder, kind of thinking in the future, is there a future in which you envision no more need for like bone marrow transplants? You can just reprogram your bone marrow from some cell and then get that yourself. Is that something that you think would be, I don't know, possible, achievable?
So that was something that since I was a postdoc, that was the final the target, right? So we have like a much more available source of stem cells for transplantation. The hurdle and the challenge that we and others face, right? So some other groups working in this field was that then the functionality of the stem cells is the hematopoietic stem cell is somewhat impaired when compared to the natural ones. So it's very hard to have completely an engraftable cell that would be then in the body and producing blood for a long time. So this has been a challenge, but it's pretty much, it's also associated with the fact that we don't have very good conditions to culture hematopoietic stem cells in vitro. So then the two things together really makes this kind of the holy grail of adult stem cell biology, to be able to generate these hematopoietic stem cells in vitro. But we are working hard on that, and not only academic groups, but also companies that start accepting the challenge. So we hopefully see some big developments in the next years.
Fascinating. I think this is a really interesting field. There's so much yet to do. Self-renewing patient stem cells, sabotaging cancer cells. That sounds all a very, very exciting work.
So something we always like to do before we wrap up is get to know the person behind the scientist a little bit. And so we always try to get a few questions for you. So the first one is to send at you is, if you were not a scientist, what would you be?
So what drove me initially to become a scientist was, so I was trying to decide whether I would become an architect or a biologist. And this was in 96, 97. There was at the time where Dolly was cloned, and I was so, I was fascinated with the concept that we could, turn one cell into a whole new organism, and that was like the balancing point to pursue a career in biology. The alternative was to be an architect, because I really I like to kind of have this creativity, and also the planning of something that you want to build. So these two things were always with me.
I would argue that now you're basically an architect of biology, building your own cells. So I would say that you kind of went both ways.
Yeah. So at the end, the two things merged. So yes, we had, like, me and my group have been architects of cell fate.
There you go. So then the second one is, what is the best piece of advice that you've ever been given? This can be professional or not, just something that's stuck with you throughout time.
So my PhD mentor, when I was finishing and leaving the lab, she told me, so when you have your own lab, so don't ask or don't do anything that you would not like to be done yourself, right? And she was meaning like, so hire or get associated with very good people, and then leave them alone in terms of give them space to be creative and to be and to contribute, and ask only for the final, keep reminding what's the objective, and don't be too much into the detail, let's say, or too controlling about the whole steps of the process, because this will be detrimental to the overall aim of what we are trying to do. And and this was something that I didn't really understand at the time, but as soon as I started my group, I really realized that this was a big piece of advice.
So don't micromanage your people, basically. Let them be.
Yeah, exactly, right. And because everyone gets their fair amount of stress and pressure at the different levels, so we all work the same, right? But it's very easy for you to become lose patience and start asking too much about and too frequently, or too much about the individual people. So you just need to remind them and to keep the goals in place, and everything eventually falls into place. Yeah.
Well, thank you again for your time. Speaking of people, is there any other announcements? Are you looking for postdocs, grad students, technicians at any point?
Yes, actually, we have an open position for a senior scientist. So this is actually a very good permanent position in the lab. So we hope to attract the best candidates from all over the world. But we are always welcoming applications for good postdocs. So either the computational biologists, but also wet lab scientists.
All right, great. Well, thank you for your time again. It's been a pleasure.
Yeah.
Thank you, Brenda and Jason. It was great having this conversation with you.
That's great to hear more about your science. Wonderful work you guys are doing. Thank you so much for talking to us.
Okay, thank you so much. Bye-bye.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests.